metabolism, and carbohydrates is going to cover understandings 4, 6 through 10, and application skills 5 through 9. So pause this video and go read through those real quickly and then come back. Okay, so first question of the day. With somebody nearby, discuss how should we study biology? We have two options here. First option is, should we look at the big picture, study the whole organism, and learn how its systems work and how the systems work and how life works by studying the entire organism? Or should we look at the parts that make the whole? Should we study all the little cells and the microscopic structures and atoms that make up that organism? Which way do you think we should study biology? Well, it turns out that there's not actually a right answer to that question. Some biologists are using an approach called the reductionist approach. The reductionist approach means you study the little itty tiny cells, and some biologists say, no, we need to study the whole living organism. So we're going to look at both of those things throughout this course. But first, we're going to take a look at something that happened a long time ago. I would tell you the year, but I'm really bad about dates. So this is a long time ago. There was this theory called vitalism, and it was basically the theory that organic molecules could not be made without the spark of life. There was something about living things that was really special, and that was the only way we could get organic molecules. Do you think that in today's age, with all the science technology that we have, that we could actually make an organic molecule from a non-living thing? I know, sounds crazy, right? Can we synthetically make organic molecules? You might be surprised if you answered no, that actually we can. And there was a guy named Frederick Wohler who artificially made urea. Urea is the stuff that's found in urine, using silver isocyanate and ammonium chloride. This was basically the first time they falsified the theory of vitalism, and so it went down in the books, and he actually has been falsified more times since then, but this was the first time. But there are some organic molecules that we cannot make even today with all the science technology we have, and one of them is hemoglobin. So if you think about it, hemoglobin is the protein in red blood cells that carries oxygen. If we could make it, we wouldn't need to do blood transfusions anymore. It would be pretty serious breakthrough, actually, if we could make hemoglobin. Okay, moving on. So there are two types of reactions. The first kind of reaction is an anabolic reaction. So we're talking about building things and breaking things apart. When we build something, we're doing an anabolic reaction. So do you remember when you were little, you probably had some Legos or some Tinker Toys or something, and you took two of them and you put them together. When you took two pieces and you put them together, you did an anabolic reaction, or sort of a symbolism of an anabolic reaction. Can you think of, pause the video, can you think of a example of anabolic reaction in life? So what is an example of when we took two smaller molecules in a living thing and build them into a bigger molecule, or maybe 5 or 10 small ones, and build them up? Pause the video real quickly and see if you can come up with any. All right, did you think about any of these things? Building proteins, building DNA, photosynthesis, or synthesis of carbohydrates like starch and cellulose and glycogen. Those are all examples of anabolic reactions. Now I have something that's kind of funny. It'll help you remember. Anabolic reaction means to build something. You see the first part of anabolic reaction has the word Anna. You guys know Anna from Frozen, right? Of course you do. What did Anna want to do? Anna wanted to build a snowman. So you can think Anna builds a snowman, and you're building bigger molecules. All right, catabolic reactions, the other kind of reaction, is when we have a bigger molecule and want to break it down. This happens when we digest food. You eat a big old piece of bread and all that starch. Your body starts to break those molecules apart into simpler molecules. That is called a catabolic reaction. Can you think of any examples other than the digestion I just told you, of catabolic reaction in the body? Pause the video and see if you can think of one. All right, did you come up with cellular respiration, glycolysis, the Krebs cycle? No? Anybody? Okay, that one was a little tougher, I know. Let's look at what these two things. Oh, here's a picture of catabolic reaction. It's like an explosion, basically, because catabolic reactions break things apart. Here we have a picture of two graphs that show you one of these is an anabolic reaction and one is a catabolic reaction. Let's see if we can figure out which one is which. I don't know if you know this, but when you take two small molecules and you put them together to build a bigger molecule, you actually have to put energy into the system. You have to add energy to that reaction to make it happen. So over here we have energy, we have our reactants, and our products over here. Now, if you look at the way this is shaped, the reactants went through a reaction and then they became products, and the final product has more energy than the reactant. So in this case, energy was added to this system. This would be an example of an anabolic reaction. We're building a bigger molecule, we have to put energy into the system. On the other hand, this graph over here, this one here shows a catabolic reaction. Here's my reactants. They undergo their reaction, but the final products have less energy than the reactants, meaning energy is released. An example of this would be cellular respiration. We break down glucose in order to release the stored energy to make ATP. So here is basically the summary of that. The anabolic reaction is endergonic. That's this one down here. The anabolic reaction is endergonic because it requires the input of energy. The catabolic reaction, on the other hand, is exergonic, and we release energy in this case. Now, IB is going to have you use the word hydrolysis when we're talking about breaking molecules apart, and the word condensation when we're talking about building bigger molecules. Dehydration synthesis is actually a term that's more commonly used in AP. All right, we'll take a little brain break here. Okay, moving on. Molecular biology. This is the branch of biology that deals with the structure and function of macromolecules. Do you guys remember macromolecules from 9th grade? You have carbohydrates, proteins, lipids, nucleic acids, all the things that every living organism is using. And we're basically only going to talk about carbohydrates today. So here they are. They're composed of carbon, hydrogen, and oxygen. Thus the name carbo, meaning carbon, and hydrogen, hydrate, which is water. So it's carbon and water. That's a carbohydrate. Now, it always happens in this, well, almost always, in this 1-to-1 ratio. This example here is C6H12O6. You probably know it, is glucose. There are a few exceptions of the 1-to-1 ratio. An example would be chitin. Chitin is a hard material that's used to make the exoskeleton of some shelled creatures. So anyway, function, we have short-term energy. Carbohydrates provide a short-term energy. They can also be energy storage. Animals store their energy, or humans anyway at least, store their energy in the form of glycogen. And plants store their energy in the form of starch. Both glycogen and starch are carbohydrates. They can be structural, if you think about the chitin I just gave you an example, or cellulose. Cellulose helps make up the cell wall of plants. So we know carbohydrates come in monosaccharides, disaccharides, and polysaccharides. Monosaccharides are a single sugar unit. So down here at the bottom, I've got all these little yellow shapes here. Each one of these is a sugar. If I hook them together, if I hook two together, I'll make a disaccharide. If I hook more than two together, I can make a polysaccharide. All right, some simple facts about sugars. Most names for sugars end in ose. Glucose, ribose, dextrose, maltose. You've probably heard all those things before. If you look at a sugar in a structure, one giveaway that it's probably a sugar is it's a ring. So here I've got a 6-carbon ring, 1, 2, 3, 4, 5, 6 carbons. One of them is sticking out. That's glucose. Ribose is a 5-carbon ring. And then I've got a 3-carbon. This one's not a ring. Glyceraldehyde is a 3-carbon chain, but still keeping true to the 1-to-1 ratio. Now, we're not going to get too much into ketones and aldehydes, but just so you know, glucose can come in a linear form. It's not usually found that way. Looks like this. When you put it together into a ring form, it changes into this. These are all interchangeable shapes. And the double bond O here can be in different places, which changes the type of sugar that it is. Both of these are C6H12O6. All right, numbered carbons. You're going to learn more about this when we study DNA, but when I'm looking at a glucose molecule, the carbon right here is called my first carbon. Now, then you can keep going around the circle. How do I know to start here? Find the O, the oxygen that's in the ring, and then you're going to move away from the little tail sticking out, and that's how you count your carbons. So I find the oxygen and move away from this tail. That's going to be carbon 1, 2, 3, 4, 5, and 6. Okay. Here are my monosaccharides, disaccharides, and polysaccharides. Example of monosaccharides: glucose, ribose, galactose, fructose. Examples of disaccharides: 2 sugar units is going to be sucrose. You know sucrose, it's actually table sugar. Sucrose is glucose and fructose together. And then you're going to have polysaccharides. There's actually 3 that you need to know: starch, glycogen, and cellulose. Okay, so let's look at how this happens. We put these 2 guys together here. We've got one glucose here and another here, and I want to join them together by condensation reaction. I want to do an anabolic reaction. Meaning I'm taking 2 smaller sugars and putting them together to make one disaccharide. See this OH here and this OH here? If I remove an H2O from these 2 molecules, they'll come out as water. And then, let me see if I can draw this on here. This H2O right here, H2O removed, and all that's left is this oxygen. Now in that case, it's going to connect the 2 sugars together. Glucose and glucose together, you'll never guess. What is this down here? Yeah, I know. Chocolate malt. This is a maltose. Glucose and glucose together makes maltose. All right. The type of link that's formed when you remove a water from 2 sugars and put them together is called a glycosidic linkage, or glycosidic bond. Okay, now let's look at glucose and fructose. Do you remember what I said glucose and fructose make up? There's the water coming out. Glucose and fructose make sucrose. Yay! Everybody likes fructose. And glucose. Okay, getting distracted. Here we go. Back on to breaking them down. Oops, I went too far. So here we have 2 sugars. They're already connected. Here's a sucrose, a glucose, and a fructose together here. And we want to break them apart. So this is going to be a catabolic reaction. I'm going to need to add water, and I'm also going to need an enzyme. Now, can they break apart without the enzyme? Maybe, but it would require a really long time or a high temperature, neither of which are available in the body. So we're going to add the water, we're going to add the enzyme, we're going to make this happen much quicker and at a lower amount of energy required for the reaction. So here we go, break them apart. It's basically the reverse of what we just learned. This is called hydrolysis. Hydrolysis is separating 2 or more molecules by adding water and enzymes to break them apart. All right, here's another picture of glucose. Here we go. you'll see in this glucose, I like to think of glucose looks kind of like a cat. I think this is like the cat's head. He's so cute, little kitty cat. And this is the cat's tail, right? And this is the cat's body. He's a really chubby cat, but we know chubby cats are cute. And he's got okay, this is actually the cat's poop right here. The cat's pooping. Cats like to poop. And then over here at the front, we have a situation. Now, this is alpha glucose. On the first carbon, the OH group is down. That's what makes it alpha. Here we have our kitty cat, our glucose molecule. There he is, his tail. He's still pooping. Okay, but on the first carbon, the OH group is up. That makes this beta glucose. Okay. So you got to be able to look at a picture and tell the difference between alpha and beta glucose because it matters when we're looking at the polysaccharides. Alpha glucose is used to make starch, and beta glucose is only used to make cellulose. Starch and cellulose. There we go. Whoops. Oh, that was supposed to be a smiley face. There we go. Okay. So, alpha glucose in starch, beta glucose in cellulose. Moving on. Now we have to realize something. All those OHs were down here at the first carbon in starch. So all the bonds that it makes when you connect them together are facing down. In cellulose, this first OH group was up. So that bond is up. And then it flips over. You see the tail is on this side now? And this OH is going to be down because that's the tail of that one. And it's going to be opposite. Every other O is up and down, and this is in cellulose. So what's happening is starch, humans have a special enzyme, and we can break all these down bonds. This is no problem. We have an enzyme for this. It's called amylase. But cellulose, on the other hand, these bonds here are up, and we don't have an enzyme that can do that shape. Now, it does exist. Some animals are able to digest cellulose, just not us. All right, here's a picture of ribose. This is actually deoxyribose. You see there's no oxygen there. D means to remove. So deoxyribose. And this is ribose, just regular ribose. Deoxyribose is found in, you guessed it, DNA. Fantastic. And regular ribose is just RNA. Okay. These are similar shapes to glucose. Can you see the difference? Here's the O. First carbon, 2, 3, 4, and 5. Yes, ribose only has 5 carbons, so the ring is a little bit different shaped. All right, here we go. Polysaccharides. Let's get these guys out of the way. We have 3 that you need to know: starch, glycogen, and cellulose. Now, each one, energy, starch, and glycogen, are both used for energy. Starch is energy storage in plants. Glycogen is energy storage in animals. Cellulose, on the other hand, cellulose is a structural feature that's used in plants for their cell wall, right? We remember that from probably 9th grade. They always like to throw in chitin. Chitin's actually not in your IB curriculum, so don't worry too much about that one. Here's a picture of what cellulose might look like. You see the up bond here and the down bond there, up here, down here. This sort of zigzag, if you're taking the test and you're not sure, and you see up, down, up, down, up, down, that's a good indication that it's cellulose. It is the most abundant organic compound on Earth. It is undigestible. You ever heard somebody say, oh, you need to get your fiber in your diet? Guess what they're talking about? Cellulose. The more plants you eat, the more cellulose you'll have in your diet. The more cellulose you have in your diet, the faster food moves through your digestive tract because the cellulose draws water into your colon. So if you have problem getting the poop out in the bathroom, you got to eat more cellulose. All right, here we go. Sorry to gross you out there for a minute. Here is starch and cellulose. You can see starch, all the connecting O's are down because these are all made of what kind of glucose? That's right, alpha glucose. Now, this one over here, that's supposed to say glucose, by the way. This is cellulose. You can see this one's up, this one's down, this one's up. We can tell this is cellulose because these are beta glucoses. Within starch, there are two structures. Amylose kind of looks like this. Pretend this is a long chain of all of those hexagons from the glucoses. And then amylopectin. Amylopectin is just like that, only it's going to be really branched like this. So humans, we eat starch and we like it and we can break it down. We break all this down. Plants, when they store starch, they can store it in these really complex structures like this, and allows them to store more sugar in a smaller